Recording. All right, good deal. Good deal. All right. So if you're looking at the chapter 13 where we just we're talking real quick about what restriction enzymes are, how we make recombinant DNA, how that leads to transgenic organisms, that kind of thing. So I just want to kind of break that down one more time. All right. So, I just hit myself in the head. So if you want to make recombinant DNA, here's the how. You got to use your restriction enzymes. We know those come from bacteria. Okay. When you cut, you make what's called blunt or sticky ends. It's just how they cut. It's kind of like using decorative scissors. Okay. You've got to then use PCR to help amplify your DNA so that you have enough to play around with. You're going to want to run a gel electrophoresis to check that your cuts were made. You're going to want to insert a reporter gene. Your reporter genes are what we call selectable markers. They are things that we like to use to ensure that our gene we wanted actually made it in there. Okay? And then we simply just allow for replication because we're usually doing this in bacteria first, and allow for replication just on the Petri dish and agar. And from there, we'll select the ones that actually got the gene inserted. All right, so again, just how do you make recombinant DNA? Use the restriction enzymes, create the cuts, amplify with PCR, go through gel electrophoresis to check that it was cut, insert a reporter gene, and then allow for replication, okay? Now, that reporter gene and stuff like that, let's kind of talk about that for just a quick sec. If you scroll through your Prezi with me. So we stopped after PCR because we watched those videos, fun times, right? If you kind of just go through the PCR, you do need to remember the PCR steps, okay? Keep those in mind. PCR, denature, anneal, extend. Denature, anneal, extend. Denature, anneal, extend. That's all you need to know, okay? Reporter genes. They can be what we call GFP, or they can be ampicillin resistance. GFP literally stands for green fluorescent protein, and then ampicillin resistance speaks for itself. I'll talk about that in just a quick sec. All right, so PCR, you have to know those 3 steps and just what they are. Just the fact that you denature the bacteria or the DNA, you add primer, that's the annealing, and then you make your DNA through the extension part. That's all that is. All right, so if you go ahead and look through, you cannot minimize this. Okay, I just got kidding. Yeah. Okay. So if you go ahead and look in your Prezi application part, this is just kind of saying, hey, so you've made recombinant DNA. Like, that's the how, right? You add a reporter gene, you do this, but like, what are you doing with that recombinant DNA now? So you have officially created what we call a transgenic organism. An organism that now has 2 sets of DNA somehow within it that it can now manipulate and use. You are ensuring that you successfully did this by adding what we call a reporter gene. So if you scroll down to the recombinant screening one, just a couple of clicks. So in the recombinant screening, what you're doing is you're adding the gene you want. Like, I want bacteria, E. coli specifically, to make human insulin for us. But I need to make sure that it's doing it. So next to that gene, when I cut open its plasmid and I insert the insulin, I'm also going to go ahead and insert a reporter gene. It's a way for us to physically, phenotypically see that it's taken it up. Because, guys, we're shooting it in there. And we're adding the enzymes. There's no guarantee that it actually is doing its job, okay? So the reporter genes, ampicillin resistance, and GFP, those are the most common ones that we like to use. So if you look at the little pictures of the Petri dishes, you're going to see the first 2 on the top. Those 2 Petri dishes do not have the DNA in them. Like, they haven't even been genetically modified. Then the bottom 2 have. So the top 2, you'll see something written LB under it. LB literally means Lura broth. It's just the agar. Don't worry. It's just, they're telling you this is just good old fashioned agar. Well, if you look, you can see the bacteria is growing. Well, that's because bacteria grows on agar. But then if you look at the next one, it says LB/AMP. What that's telling you now is they've added the antibiotic ampicillin to the agar. But there's no bacteria growing. Well, that's because this bacteria isn't resistant because it didn't take up that DNA. So then the bottom two, now you have the ampicillin resistant bacteria growing because you can see the little colonies on it. And then the one right next to it, they added something else that activates the other gene for the GFP. So not only is it growing, but now it's also glowing in the dark. So it's kind of like, what? Ooh, my cord. Ooh. My puppy ate my charger cord, and she kind of actually made it through the coating, and I haven't found my electrical tape. I'm waiting to set my house on fire. I swear. It's awesome. Okay. So anyways, this question, actually, the test review I gave you yesterday, if you look on the back, I gave you some practice questions. There's some questions that actually show this same picture. And it's just asking you, like, do you understand what's going on in this picture? So definitely go ahead and look at that one. Actually, go ahead and get your test reviews out, and I'll go through that question with you real quick. So get your test review from yesterday out. If you're missing any, I think if you don't have it, it's on my, I think I have some extras sitting there on my desk or in my black binder with the class period, and I'll pull up the test review as well. All right, any questions so far? I was still eating my breakfast, so if you don't mind. Okay, doing this video conference makes my computer run super slow. Like, like I'll be lucky to open it at this point. Keaton says no questions. All right, good. Okay, so go ahead and look at that test review real quick. Look at the back where the practice questions are. Let's go ahead. All right, so if I asked you the how, if I asked you, hey, how are you making recombinant DNA? You need to be able to tell me the basics. Using restriction enzymes, making cuts, amplifying, gels, reporter genes. You need to be able to tell me that. Okay. Oh, I can hear Presley in there coughing. Her fever's still over 100. She ate a couple of bites of taco last night, but other than that, she's still down for the count. All right, so let me go down. Like I said, let me get to those questions. All right. Come on. Okay, so look at question. It's question 32 on my side. I'm pretty sure it should be the same one. Making sure I'm looking at the right one. So it has the pictures of Petri dishes. And it's telling you, hey, I added stuff to this. I didn't add to this. Da, 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 da. And then it's just asking you, if no new mutations occur, it would be most reasonable to expect bacterial growth on which of the following plates? Okay. So if you didn't induce or change any kind of mutations or anything, right? Actually, go up. Sorry. You need to look at 31. I don't know why I have that worded numbered. Sorry. Right above, it gives you the explanation of what's going on. You're working with the lac operon. It says that one of them has functional, you used restriction enzymes, da, da, da. You added high concentrations of this recombinant DNA, and your bacterial growth medium has glucose in it. So you're streaking the plates down at the bottom. So if you look at the first column, it says your it's glucose. So number 1 and number 4 both have glucose. Number 2 and number 5 now have glucose and ampicillin. And number 3 and 6 have glucose, ampicillin, and lactose. All right. So it's telling you that they've modified it to where they have fixed the lac operon because it was non-functional up at the top. So the first row says that you've added the plasmid. So you added the functional correct operon gene. So in plates 1, 2, and 3, those bacteria growing have the plasmid. But then it says the second row, these bacteria do not have the plasmid. So plates 4, 5, and 6 have bacteria on them, but the bacteria don't have the plasmid to give them the functional gene to break down lactose. And in this case, since it says ampicillin is also on the Petri dish, that's telling us that ampicillin resistance is our reporter gene. So the question says, if nothing new is added, no additional mutations happened, which plates would you expect bacteria to grow on? Well, so bacteria need to grow on glucose no matter what. Like bacteria love glucose, so they're going to grow if there's glucose. So plate number 1 and 4 both work. Plasmid or no plasmid, they love glucose. But on plate 2 and 5, it has ampicillin. So you can only grow if you have the plasmid. So only plate number 2. And then plate number 3 and 6, again, you have ampicillin and lactose. So you have to have the plasmid. So your correct answer on that would be E. All right. Okay. My dog is clawing at the door and I'm about to go psycho. No! Okay. I'm in the living room. If the sign, and then go ahead and look at the last question. Or actually, look at the one above it, number 34, real quick. Look at number 34. It said, if the scientists had forgotten to use DNA ligase, think about what DNA ligase does. Ligase for links. It's the one that brings the DNA back together. If they forgot to do that when they were making the recombinant plasmid, so you cut DNA, you added, but you got to link them. And that's where that enzyme comes in. Bacterial growth would most likely have occurred on which ones? So if you forgot to link the new gene, like you did not add DNA ligase, then that new gene is just going to go whoop and fall back out. So naturally, if there's no gene that actually makes its way into the plasmid of the bacteria, then you're only going to see growth on the only plates that it could survive naturally. And so that's only going to be plates number 1 and 4 only. The answer would be B. All right. Puppy hair. All right, look at the last one. So if you use the cultures to perform another experiment, to the right, okay, so there's another one, using medium containing lactose. So now lactose, no glucose at all. Growth would occur on which of the following? So in this case, you've had to insert a functional gene that can break down lactose because it was non-functional according to the paragraph above. So in this case, you're only going to see growth on plates 7 and 8 because the plasmid had a reporter gene to help it grow with ampicillin as well. So that's why it gets to grow on number 8. But 9 and 10 don't have the plasmid, so no growth there. All right, kind of go back to your Prezi real quick for me. You can put your test review off to the side. We'll look at more questions tomorrow. All right, so again, like the question comes down to what reasons, actually, I'm skipping a couple of clicks. So I'm on the little part that says gene library after the reporter gene. No! The gene libraries, guys, it's exactly what it sounds like. You have separated DNA, you've worked with it, you maybe even have already recombined it. And what do? You make a lot of it. And you keep it in basically these little trays in a fridge. You've literally made a library so that if somebody else, hold on, my puppy is scratching at my door. No. No. Okay. Sorry. Oh, okay. Anyways, so gene libraries, they legit are just what they sound like. If I'm running an experiment and you have ampicillin resistant lactose plasmid already, I can just go and be like, hey, I need some of that. Thanks. Whoop. That's all that is. Okay. Continuing. I'm clicking. That's all. Gene library is just a term. Okay. Complementary DNA, we already kind of mentioned that, skipped complementary. After complementary DNA was this really neat little picture. It looks like a laser gun. Pew, pew. Okay. That is actually something called a gene gun. Let me thinking about it real quick. I wanted to double check how fast it does it. I totally forgot. It's like so fast. All right, so here's the thing. One method that they use to actually, so gene library, right? They have their DNA, right? Here it is. Woo! They have this plasmid already created. It's ready to go. It's recombinant DNA, and they want to quickly put it into a bacterial cell. So they grow their bacteria on a petri dish, basically, and there's multiple ones. Some of them will put them in it looks like a little miniature oven, but in that oven are these gene guns like that, and they pew, 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 and they shoot these little pellets, okay? And these little pellets have the plasmids in them, but they are shooting them at, let me see, I think it's like 800 PSI. 800 PSI, 30 RPM, something like that. Like it's super fast. I was trying to see, I want to say it's something like that. Like 800 PSI, something. Anyways. And so they just literally just shoot it into the cell. So it's going so fast, it breaks through, but it's like so quick and it gets into the cell fast. So that's just something fun. I just wanted to share it with you. It's a gene gun. How did we know? So what can we do with this recombinant DNA after we made it? What are we doing with these transgenic organisms? Well, first thing is gene therapy, guys. That's where we're at, right? I had you look at that article last week. Whether or not you agree with it or don't, it's what's happening today. What have we been able to do with gene therapy? People who are paraplegic are starting to walk. People who didn't have feeling or sensation due to nerve damage are now feeling in those areas. Gene therapy itself is also, we're looking at applying it towards genetic diseases. If we can get to the cells that are affected early enough before they degenerate or whatever and infect them with the healthy genes, then those healthy genes can start to take over. Okay, this is kind of a 21st century stuff, but we already were doing something similar whenever we were doing stem cell transplants anyways. Like the man in Germany back in 2007, back in 2007, he had AIDS or HIV. He had HIV and he had leukemia. Not only do you not have an immune system, but you also don't even make proper white blood cells. Like, what? So back then, they gave him a stem cell transplant, essentially gene therapy without recombinant DNA. They took the DNA, or the stem cells, sorry. They took cells from individuals that have a mutation, right? And they took the mutation, injected him with that bone marrow, and allowed it to take over his bone marrow count. Because what happens is 1%, it's probably actually higher now. I haven't checked it recently, but approximately 1% of the human population has a mutation on your, let me draw it, on your T cells. So your T cells should have some kind of like dock. Sorry, I can't see the screen. They should have some kind of like docking station. And that's how HIV infects you, because it has a dock. Well, 1% of the human population, your T cells don't have that dock. They don't have that protein. So without that protein, HIV can infect. So they gave the man a bone marrow transplant, which is what you do when you have leukemia anyways. But instead of them just giving him some random transplant, they specifically gave him the one with the mutation. So in 2011, 4 years later, they reported finally that he was HIV free. So we've been doing different versions of gene therapy for a while. Okay. All right, I'm going back to the Prezi, just kind of clicking through what are some other examples we've done with recombinant DNA. What other transgenic things have we done? We have goats that their milk now make our blood clotting factors. Like if you're hemophilic, if you go in for surgery and you start bleeding out, they're giving you clotting factors made from a goat. Yeah. We've made food for third world countries. It's called golden rice. That's kind of the main one, but it's not the only one now. It's infused with vitamin A and stuff because soil content in different areas doesn't have certain minerals that plants can actually start to synthesize different vitamins. So we've created this vitamin-rich plant, and it can grow anywhere and still have everything that you need. So we've done that. I'm sure if you think about it, there's a lot of things you can think about that we've used transgenic organisms, right? E. coli for our insulin. Just some of our plants, not a lot. People go overboard. They think everything you eat or drink or come in contact with is transgenic. No, it's not. Ugh. On the market, there's actually only like 7, maybe 7 or 11, like some, I forget. There's very few actual foods that have been approved that are actually out on the market that you eat. Where people get all crazy thinking it's everywhere is because when you're buying processed food, when you're buying that thing of pasta, that mac and cheese, that cereal, yeah, it has genetically modified or transgenic organisms in it because of the wheat. Wheat is the most common transgenic organism out there. So yeah, if you're buying processed food, you're eating transgenic. Go to the produce. I think the only produce we have are tomatoes, and even then it's only certain brands of tomatoes. Tomatoes, some zucchini, dog hair on that stuff. Zucchini, that, I feel like that's it. I think that's it. Oh, my husband's texting me. So yeah, you want to avoid transgenic, just buy natural. And guys, here's the difference. There's a difference between something being transgenic and something being polyploidy or selectively bred. Transgenic is we actually have modified it in a lab, like, right? Where we've actually done something to its DNA, like flounder DNA in tomatoes to make them kind of anti-freezing, okay? But we've selectively bred strawberries. We've selectively bred apples. We've selectively bred bananas. They are still bananas. They're still apples. Like nothing changed with their DNA. They don't have anything in them. It's just we made it to where, ooh, this apple tree seemed to make bigger apples. And look, there's like barely any seeds. I like that. I'm going to keep growing that tree over and over and over. Eventually, that's the one that's the most common, okay? If you're ever curious and you want to look up the fun history of bananas, that's actually kind of interesting. The bananas we eat today are not the bananas we ate 50 years ago. That's all due to a virus that got to a particular species of banana that we used to eat. And so they had to selectively breed for one that the virus couldn't attack anymore. So the banana you eat today is not the banana your grandparents ate. So kind of interesting. So be able to give me some examples of how transgenic organisms are in our environment. But then also think about what does this mean, right? Like, what does that mean for you? Transgenic organisms, is it a good? Is it a bad? Where do you fall on this? Where do you fall on this line of, yes, we need GMO. No, we don't. GMO crops are good. GMO crops are bad, but be able to back it up. Like, what are some reasons? Remember, guys, when it comes to backing up stuff, especially on an FRQ or something, you got to give me some kind of legitimate reason, right? So we've made genetically modified organisms for things such as, well, right, the flavor savor tomato, so it doesn't freeze. So if a crop has had a really bad winter, those tomato plants are still going to be good, right? That sounds great. But then we've also modified things to where they grow and they grow and they grow and they grow, and there's no pesticides. Why? Because they have natural pesticides. Well, that means that anything that eats it either dies or doesn't come back. What does that potentially have on the big picture? Like, how could that potentially affect our ecosystem, having all of these crops? I was looking at an article last night. Scientists are really, really concerned. Our insects are dying at a huge rate. Like, they're ready to call it the 6th mass extinction. Like, really big. So curious how that's going to affect. So just kind of think about how GMOs could affect us on a large scale. The next little clips, here's the thing. There's something called RFLP that I have on there. I actually need to take that off just because I don't ask about it anymore, because I ask about STRs. RFLPs are what we used to use when we were DNA fingerprinting. Not anymore. We use STRs. The Innocence Project, all that kind of stuff. So I'm skipping through all that. We already looked at SNPs once before. I'm not going to focus on that. And then the last little bit was just about ethics, like, where do you stand, right? Did you hear the thing about Coke? Gosh, I have to look it up again. Oh, apparently, and I don't know, I heard this on the radio the other day. I was like, I haven't looked into it, but apparently there's some kind of chemical in Coke that they're finally taking out because it's actually used in flame retardant. I'm like, well, that's nice. Okay. Just think about where you are in this world of gene therapy, genetics, transgenic, all that kind of stuff. At what point do you consent to your stuff being used? I know somebody asked me yesterday. So I'm going to be bone marrow, like I'm swabbing to be put in the system for Be the Match, just because, if I could help, I want to help. And so somebody else is doing that. And they asked me yesterday, and they're like, hey, so how does that work? Like, is my stuff in the system forever? And yeah, technically it is, guys. If you do 23andMe or Ancestry, your DNA and everything's in the system. So at certain points, yeah, your stuff can be used. Like, you kind of gave your consent away, whenever you sent that stuff in. A company in Europe years ago wanted to actually start what did they call it? Human DNA library thing, where they wanted people to donate their DNA. Just a saliva, you're right. Nothing big. You didn't have to do anything. But what you were basically signing away was that you were giving consent for them to have access to your DNA and do whatever they wanted to. And it never got through because one of the things in their paperwork specifically said cloning potentials for future stuff. And people are like, no, I don't want another one of me walking around potentially. So just kind of that last little part about the ethics, just kind of look at it and be like, where do you stand? Monsanto, right? Dun, dun, dun. That's that big one. They're our big GMO company. They're not the only ones, though. Have they done great things? Yes, they have. Have they done questionable things? Yes, they have. Where do we stand now? Well, I don't know with the change of political arrangements now, but I know under Obama, the Monsanto lead scientist was now over the FDA, and one of Monsanto's lawyers was now part of Attorney General, Supreme Court, I don't know, something like that. So that company definitely has some reach. Is that good, bad? I don't know. What they're doing is some great stuff. As long as they stay legal, I guess. That's where you get into this ethics situation. So, anyways, any questions I can answer for you. We'll look at, I don't want to take up, you'll have 15 minutes. The last little bit that I want to look at tomorrow is the polarity stuff, and I'll be in class tomorrow. My husband will stay home with Presley tomorrow. So we'll look at the genes and development, chapter 14 tomorrow. What I would like you to do for the remainder of class time is go ahead and start looking at your test review so that tomorrow, after I kind of brief you on the polarity stuff, if you have questions on the test review, I can answer them. Okay. And I'll talk to you about your FRQ. So please come in tomorrow, quick, get settled so we can get everything done in enough time. Okay. Any questions, John? Yes, no, no, yes. I'm sure he's sitting there with his headphones in, not even paying attention, right? Yeah. Either that or he probably has his head down. Mm-hmm. Mm-hmm. Am I right? Okay. All right. I'm assuming Keaton says no questions. Okay. All right, good. Yeah. So he has his headphones in, so he's not even listening, right? All right. Okay, guys. I'll see you tomorrow. Bye. I've got it.